This is my presentation on immunological assays and different lab techniques that are commonly used in immunology. So, immunological techniques are used in multiple settings, from laboratories to clinical settings. And many of these techniques are based on the use of antibodies because they can detect, purify, and quantify antigens. Since antibodies can be produced against any type of macromolecule, these antibody-based techniques may be used to study any type of molecule in a solution in cells. And with these methods for producing monoclonal antibodies, it's greatly increased our ability to generate antibodies of almost any specificity. And this helps us gain a better understanding of our immune system and its diseases. So, the first technique that caught my attention was the flow cytometry. By staining cells with fluorescently labeled probes specific for those molecules and measuring the quantity of fluorescence emitted by the cells, the lineage, maturation, or the activation status can be determined by analyzing the expression of different cell surface or intracellular molecules. It can be used for immunophenotyping, cell counting, and cell sorting. The principle is that it analyzes cells in fluid suspension based on their physical and chemical characteristics by having cells labeled with fluorescent antibodies pass through laser beams for analysis of fluorescent emissions and scatter. And this can be applied in immunophenotyping, cell counting, and cell sorting. And clinically, it can be used to diagnose leukemia, lymphoma, and the progression of HIV. A second technique I found very interesting was the enzyme-linked immunosorbent assay, or it's also called ELISA. It utilizes antibodies and enzyme reactions for the detection of specific antigens. The antigen or the antibody is covalently coupled to an enzyme and quantified by determining with a spectrophotometer at the rate which the enzyme converts a clear substrate to a colored product. Antibodies immobilized on a solid surface capture this antigen, bind substrate reactions, and produce a detectable signal. It can be used in diagnostic and research for protein quantification and cytokine detection. The most commonly used variation of this is called the sandwich variation, which measures the antigen between two layers of antibodies. The target antigen must contain at least two antigenic sites capable of binding to antibodies. Monoclonal or polyclonal antibodies can be used as the capture and detection of antibodies in the sandwich ELISA systems. And finally, the last technique that I was drawn to was the immunoprecipitation. It relies on the specificity of the antigen-antibody interactions to isolate and purify the target proteins from a biological sample. The antibody specificity for one protein antigen in a mixture of proteins is used to identify the specific antigen. And it does this by having the antibody added to a protein mixture, which is typically a detergent lysate. The A or G proteins that are covalently attached to the beads is added into the mixture. The sample then containing the protein of interest is incubated with specific antibodies that recognize and bind to this target protein. Then the antibody protein complexes are immobilized on a solid support through non-covalent interactions. And then finally, the eluted protein can be analyzed using many techniques like water blotting, mass spectrometry, and enzyme activity assays. This technique can be used in protein-protein interaction studies and drug target validation. And in a clinical setting, it can be used to investigate disease mechanisms, the discovery of biomarkers, and the drug development. And here's a little image of how the immunoprecipitation kind of works. Some modern innovations of these specific techniques that I chose. So for flow cytometry, It's the use of heavy metal ions instead of the fluorochromes, which allows for a high-dimensional analysis of cellular phenotypes. The ELISA has the multiplex ELISA and the digital ELISA. So the multiplex detects the multiple analytes in a single sample, which simultaneously enhances the efficiency. And the digital one uses digital readouts for ultrasensitive quantification of analytes, which improves the detection limits. The immunoprecipitation has the chromatin immunoprecipitation, which is used to analyze the protein-DNA interactions, and it provides insight into the gene regulation and chromatin dynamics. It also has cross-linking immunoprecipitation, which enables the identification of RNA-binding protein targets that aid in the understanding of RNA-protein interactions. And here are the sources I used. Thank you.